This is a relatively new experience for me, but hello out there in YouTube land. My name is Matthew Avison. If you don't already know who I am, I'm a senior lecturer in the School of Cellular and Molecular Medicine, University of Bristol, and I've been asked by Bristol Bridge AMR, which is a research consortium at the university funded by the EPSRC, to give you this crossfire event, which is about antimicrobial resistance and about some of the work that we're doing, some of the work that you might consider doing if you're interested in this area. The aim really is to give something that's relatively accessible to everyone, but it's particularly aimed more at the kind of physical sciences, chemical and biological sciences, really, in half an hour, with a title of, you know, we're all in this together. It was impossible for me to really go into too much detail about everything. And so, with apologies to my colleagues in the social sciences, particularly where I think their research is particularly important for AMR, understanding behavior and how behavior affects usage, for example, of antimicrobials. I think that's really important, but I'm not going to talk about that today because I don't have time. The other thing to say is I'm going to go through the presentation. There's some slides to look at. You won't have to look at me. You'll be pleased to know all the time. But if you've got any questions, if you type those in, and when we get to the end, hopefully I'll have some time to answer those questions. And if I don't get round to answering them online, then I'll make sure that we do some written answers so that they're available for you to read later on. So sit back and get ready for your questions. Right, I have to press a couple of buttons to get my slides on. That one, and then that one, and then that one. Okay. So the aim really is to give you an overview of how many different disciplines can work separately and also together to combat the problem of antimicrobial resistance. I want to start off by briefly talking about antibiotic resistance in general. Many people are familiar with the idea that antibiotics have been around for an awfully long time. So I was born in 1973. Most antibiotic classes that are used today were actually discovered before I was born. Now, they were discovered. These are molecules which are naturally produced. They're natural products. They're produced by bacteria and by fungi in the environment. And they've been in the environment for millions and millions of years. They're produced as a way of microbes in the environment basically competing for space and killing their competitors. And all we've done really is we've turned those natural products into a way of killing bacteria in an infection. Effectively, we've made them so that we can use them at very high concentrations, much higher than are found in the environment. But nonetheless, these are ancient molecules. They've been around for an awfully long time. Now, after I was born, very little has been done in terms of discovering and developing new antimicrobials, partly because the drug companies felt that they'd discovered enough. There wasn't really a market for anything else, and partly because most of those really easy to discover antimicrobials, the ones that are in people's back gardens and are all around us in the soil, they'd already been discovered. If you like, the low-hanging fruit had already been picked. So it's been more difficult to generate new antimicrobials going forward, and we'll talk about that a little bit later on. At the same time as there's been a reduction in the amount of antimicrobials being developed, antibiotic resistance has become a real problem. And this is a global problem. In the EU, there's a significant number of deaths per year attributed simply to antimicrobial resistance. But the same is true, according to World Health Organization data, across the world. It's a global problem. The specifics of the problem are different in different countries. So, for example, in the rich Western countries, we're really talking about healthcare-associated infections. People getting infections really from the bacteria that live on them or in them. But in other parts of the world, particularly in developing countries, antibiotic resistance is disproportionately felt when we're talking about communicable diseases, things like diarrheal diseases, TB, pneumonia, these sorts of things. And of course, they're the parts of the world where access to antimicrobials is very much limited by poverty. And so there are particular issues around antibiotic resistance there. Antibiotic resistance in bacteria is something that can be multifactorial. Bacteria can use multiple mechanisms, but really there are two mechanisms that are particularly important for us to understand. The first is degradation of antibiotics. So this is a picture of an enzyme, a little machine that's produced by a bacterium. And what the role of this machine is to destroy antimicrobials. Some of the machines won't destroy them, but instead they'll modify them in some way. And I would say that the majority of antibiotic resistance is caused by these enzymes being produced by bacteria. Will have taken an awfully long time to evolve. It probably takes about, I don't know, maybe several million years to evolve an enzyme to be able to do this job. Clearly, this hasn't happened since we started using antibiotics clinically in the 1940s and beyond. So really, we need to understand where these things have come from. Where have these enzymes come from? And they've come from the environment, really, predominantly. They've come from the fact that environmental bacteria have been in the presence of antibiotics for millions and millions of years. As I said, antibiotics are natural products. And so these environmental bacteria have had plenty of time to evolve these mechanisms, these enzymes. And those enzymes are encoded on DNA. And that DNA, because it's bacterial DNA, can be passed relatively easily from one bacterium to another. They do this by various mechanisms, which I won't go into now, but the predominant, most important mechanism of transfer for antibiotic resistance is the plasmid, a circle of DNA moving from one organism into another. And this can happen across species. So if you think about it, all of the antibiotic classes that we use tend to be found in soil or in water, in the environment around human habitation, really. That's where the antibiotic resistance will have evolved as well. So antibiotic resistance genes, these plasmids, are going to be found in the environment. So as we interact with the environment, potentially we can pick up these antibiotic resistant bacteria and their genes, and that can be shared with our own bacterial flora, and therefore potentially drive resistance. So that's the predominant way by which antibiotic resistance evolves, through these enzyme-mediated mechanisms. The other mechanism that I just want to briefly mention, because it relates to some work that I'm going to talk about later, is the idea that bacteria can have a reduced permeability to antibiotics. They can actually block the entry of antibiotics. This is particularly a property of what we call the gram-negative bacteria. So these are bacteria which have got an extra outer membrane. So it shows up green here on the slide. This is a lipid bilayer. It's made of an oily substance, and most antibiotics are water soluble. And anything water or water soluble can't mix with this oily layer, so it can't pass through the oily layer. Instead, for anything water soluble to get into these bacteria, they need to get through little protein channels called porins. And these protein channels can be reduced in their abundance by bacteria. Or alternatively, the bacteria can produce pumps, which actually pump the antibiotics back out as well. So particularly for gram-negative bacteria, antibiotic resistance can be driven by this reduction in the ability of antibiotics to enter the cell, this reduced permeability, as we call it. So they're basically the main and most important mechanisms of resistance that we need to consider for the purposes of this brief talk, at least. But now I just want to talk about what research can do to help the problem. And I'm really focusing this around on people from all sorts of different disciplines. So hopefully you out there listening to this now. And of course, one of the first things we need to consider is it possible to identify new antibiotics? Now, antibiotics probably will work best if they come from natural sources. As I've explained, most antibiotics that we currently use are derived, at least, from chemicals which are naturally produced in the environment. But really, if we want to discover new antibiotics, we want to look in weirder and weirder places. We want to go all around the world and try and find them in strange places. There's two reasons for that. One, if we go to a deep sea thermal vent, like on the slide here, this thing will probably have some bacteria down there. If there are bacteria, the chances are those bacteria will produce antibiotics as a competition mechanism. However, there will also be antibiotic resistance probably that's evolved in this environment. But because people are very unlikely to go down to the bottom of the sea, to these deep sea thermal vents, it might be less likely that the antibiotic resistance that's in this environment will end up in your skin bacteria or your gut bacteria, slowing down the evolution of resistance to these novel antibiotics that you find there. Of course, the other reason for going down there and looking is that nobody's done it before, so you might find something new. There are all sorts of other places that we can look as well. So down in caves where various bacteria and other microbes might live, but also in the soil as well. When we've looked at developing antibiotics in the 50s, 60s and 70s, all we looked at were bacteria that we could grow in the lab. And actually the vast majority of bacteria and fungi that grow in soil can't grow in the lab. And so actually we've missed probably 90% of all the microbes that live in the environment, and we've never even tested to see whether or not they do produce antibiotics or not. So maybe we need better ways of trying to grow these microbes in the lab. And this iChip is something that was published a couple of years ago in Nature as an example of a device which could be used to potentially enhance the growth of environmental bacteria. So if you're going out on a field trip somewhere and you're going to some strange place, maybe you're collecting some biological samples or something, maybe you might want to consider whether or not we could potentially collect some samples to look at antibiotic production. So for example, we're working with an earth scientist at the moment on a project where Kate Hendry in the University of Bristol is going down and picking out deep sea sponges. These are coming from about 2 kilometers down under the ocean. And in these deep sea sponges, there are bacteria. And in those bacteria, there may well be antibiotics that are produced. So she brought the sponges back for a particular purpose, but we're now working with her to isolate bacteria from the sponges and potentially to identify novel antibiotics from that. There may be other examples. But it's not just the environmental scientists. Maybe the biological scientists amongst you would be interested in some animals. For example, ants have got bacteria that live in their gut. These are competitive environments, and so within there, they potentially produce antibiotics. And there's some work going on at other universities where they're looking at these particular bacteria that live in there. Some other animals produce antibiotics themselves. So this is a toad. If you've ever seen a dead toad, you'll probably realize that the only thing that's left after a while is its skin. Everything else rots away. And the reason why the skin doesn't rot away is because it's full of antibiotics. Antimicrobial peptides are present in this skin. And one of these antimicrobial peptides was developed up by a colleague of mine in biochemistry, Chris Dempsey, and actually a very similar product is now going into phase 3 clinical trials as a topical ointment for treatment of skin infections. The other thing to point out, of course, is fungi are an incredibly good source of antibiotics. The reason for that is that fungi are eukaryotic organisms like humans are, and so they tend to produce antibiotics that don't impact on eukaryotic organisms. So they tend to be quite specific for bacteria, killing bacteria. So side effects tend to be less for fungal natural metabolites. Now that's a gross generalization really, but it'd be very interesting to see whether we can identify natural products from fungi. And my colleagues in biological sciences, Andy Bailey and Gary Foster, are leading a team, natural product chemists at the university, and myself and Jim Spencer in the more microbiology end to test these compounds that we're generating from fungal genomes really, because one of the problems with fungi is they really don't grow very well in the lab, but if you can get the genes out that might encode these natural products, then you can potentially express those genes in some heterologous host, and actually they're using a fungus that makes soy sauce, so you can grow it in a big vat. You can put the genes in and they can produce, instead of soy sauce, you can tap it off at the bottom and you might find you've got a new antibiotic. So that's the hope with that sort of project. Once you've got these biosynthetic pathways, then the synthetic chemists can come in, the synthetic biologists. They can really get in there and potentially mix and match the genes that are involved in this biosynthetic pathway. So this is a little bit like a car production line. You've got the chemistry being directed by various enzyme machines within the organism that's producing the natural product. like the old Model T Ford, at the end of this production line, all you've got is a single car in a single colour with a single engine. Of course, nowadays we know that you can get a car in any colour with any engine, and you can choose the upholstery as well. And the way you do that is because production lines in modern car plants, there's variation at each stage. You can introduce changes into the structures of these machines, and so you can end up with variation. So synthetic biology could potentially give us a wider range of end product. And instead of having one antibiotic that might not be particularly good, we could generate a whole raft of different antibiotics. And hopefully one of those or two of those might be better than the existing type. There are other ways we can improve antibiotics. So as I explained, many of the reasons why antibiotics fail, particularly against gram-negative bacteria, is that they just don't get into the cells very well. So they don't actually get through the envelope because of the permeability barrier. So we've been working with some chemists and tried to modify an antibiotic which has failed and make it so it gets into bacteria more easily. So for example, the black molecule on this slide here is an antibiotic called lactivicin. Now lactivicin is an antibiotic, it's a natural product, but it's useless really against gram-negative bacteria because it doesn't get into the cells. What our chemistry colleagues have done is they've added this red chemical to it. This is a siderophore. Siderophores are chemicals that are produced by bacteria. And they're produced by bacteria for a purpose, and that purpose is to go and scavenge for iron in the environment. So the siderophore is produced by the bacterium. It goes out, scavenges for iron, and then the bacterium pulls the siderophore back into the cell so it can get hold of the iron. Bacteria need iron, and there's not much iron available to them. So by joining the siderophore to the antibiotic, we've been able to trick the antibiotic, trick the bacterium into taking up the siderophore conjugated antibiotic, pulling the whole thing into the cell, and a little bit like pulling the Trojan horse into the fort. All of a sudden, all the Greeks get out with their swords and start killing. And that's what the antibiotic does. So with this method, we've been able to increase the potency of this lactivicin by about 1,000-fold against some incredibly resistant bacteria. Bound to be other chemical modifications that you could potentially make that would increase the uptake of antibiotics, potentially through sugar transporters or through other methods. Another way of doing it is to consider the idea of using nanoparticle delivery systems. This is something that could potentially be useful. What we're really doing here is we're basically taking an antibiotic and increasing the local concentration of that antibiotic at the surface of the cell by delivering the antibiotic to the surface in some way. So this is with a silver particle delivery system, but there may be other delivery systems that can be used to bind the antibiotic effectively to the surface of the cell, effectively concentrating it and getting it into the cell much more easily. There's a whole range of examples of that. When we think about what physicists or engineers might help, I was thinking, of course, they could do lots of other things, but one of the things that we're particularly interested in is the idea of development of new diagnostics. We know that antibiotic resistance develops because we overuse antibiotics. We use them inappropriately. And one of the main reasons for that is in healthcare, we don't have very rapid and very good diagnostic tests that allow clinicians to make very quick decisions about whether to use antibiotics or not. And of course, therefore, they're likely to err on the side of caution. So what we're really thinking about in terms of primary care, in terms of GPs, is whether or not a bacterial infection is present or not. Because if it is, you perhaps need an antibiotic, though even then, not always. But if a bacterium is not causing the infection, then clearly an antibiotic is useless. So what we really want is physical machines which can go in there and can actually detect very small numbers of bacteria, hard to see, in a complex background. In contrast, there are some bacterial infections where it's much more about giving the correct antibiotic rather than whether an antibiotic is necessary at all. So in sepsis, if you give the wrong antibiotic, for every hour that the patient's on an antibiotic that doesn't work, their mortality rate will increase by 7%. And so really what's happening out there is clinicians are increasingly using last resort antibiotics. They're worried that if they have the wrong antibiotic, then their patient might get worse and might die. Now, what would be great is if we could speed up diagnostic testing for them to know whether or not an antibiotic is, which antibiotic is the one that's required. At the moment, it can take several days to actually get that result back. So we really want ideas of looking at very small numbers of bacteria in complex systems like blood samples, where there may only be one bacterium in every milliliter of blood. It's very technically demanding. Finally, when we're thinking about what physical scientists might get involved with in this problem, I'm thinking about things like mathematical modeling. We know that many antibiotic resistance mechanisms are transferred via plasmids. I've explained this already. But not all antibiotic resistance mechanisms are as successful as others. They don't move around so quickly as others. We don't really understand why that is. It may be to do with transfer rate, and that might be to do with contact between the organisms. It might be to do with how rapidly the DNA moves between organisms. It might be to do with the cost that carrying that plasmid has on the new organism. It may be that it's very complex, that in the environment it works in one way, but in the human gut maybe it works in another way. What I'm interested in is potentially trying to parameterize this problem, to try and maybe in the lab or some other kind of in vitro setting to put parameters on all of these and then have mathematical models develop which can be tested to ultimately predict the behavior of antibiotic resistance and how it spreads. And then we can really focus our attentions on surveying resistance to certain antimicrobials, whilst other resistance mechanisms we might not worry about so much. Of course, the same thing applies to infection more generally. Infection is about transfer, really, from one person to another or from one body site to another. And so there's an awful lot of modeling that can be done to look at where we might make interventions to reduce the numbers of infections. Of course, if we reduce the number of infections, then we reduce the need for antibiotics, and then ultimately that will reduce the rate by which antibiotic resistance spreads. So that summarizes everything that I wanted to say about the physical sciences predominantly. And the reason why I focused on that is because the work that we're highlighting here is really funded through the EPSRC's funding of the Bristol Bridge AMR Consortium, which is a 2-year project which finishes next September. And the idea is that we will bridge the gaps between engineering and physical sciences and antibiotic resistance researchers. If we can set, as antibiotic resistance researchers, if we can set the questions, then our physical science colleagues can potentially come up with those clever, innovative solutions, and we can work together to take those solutions forward, potentially by using our clinical colleagues to work towards putting them into clinical practice. Bridging the Gaps consortia funded across the UK. So wherever you're listening to this from, there may be one close to you. If you're interested in ours, then the website is there. And I'll really finish off my talk just by acknowledging my own funding, which has come from the UK Research Council's cross-council phase 1 AMR consortium calls. Through AMR Thing 2, we've got one project that I lead on and another one which I've talked about today on fungal metabolites, led by Andy Bailey in biological sciences. And then I have a project looking at transmission of antimicrobial resistance from the environment into people, which I'm the lead on, which is funded through predominantly NERC, but also I want to acknowledge support from the MRC and the BBSRC. Okay. Thanks, Sabine, for organising this. Thank you to everyone out there who's been watching, possibly no one, but if you're there, thanks a lot. And remember, think about antibiotic resistance, think about you being part of the problem, but also potentially being part of the solution. Bye.